In this series of video seminars, I will describe and explain our recent findings that mitochondria in cells are far hotter than the cellular environment in which they reside and than we once supposed. I'll then switch gear to consider the possible ramifications of those findings and potential objections that have been raised to the conclusions that we've drawn based on experimental findings. At the outset, I want to emphasize two aspects. First, that our work remains controversial because of its paradigm-shifting nature. In part 8 of this series, I'll address some of the specific criticisms of our work that have been expressed. While we have validated our findings using two independent methods, we remain open to the possibility that our conclusions are erroneous. Time will tell, and I hope that these videos will stimulate debate and further experimentation. Second, much of what I will present in the second half of this series is pure speculation, albeit based on actual published data. Plausible arguments that may or may not prove valid in the longer term, but which, once again, I hope, will stimulate the field. I've titled this initial segment, "The Elephant in the Lab," after the title of our recent paper in TIBS, which considered why mitochondrial heat production has for so long been ignored. When we set out on this study, we ourselves were guilty of this same rather narrow thinking, like everyone else in the field, and even high school students in most countries. We considered mitochondria as being primarily geared to the supply of energy to the cell in the form of ATP. At least this applies in differentiated cells. Over the past couple of decades, it's become clear, however, that mitochondria are also key cellular organelles in many aspects of metabolism, cell signaling, immunity, apoptosis, and ionic homeostasis, especially of calcium. But the mitochondria were also a source of heat was rarely considered, though even when it was, the steady-state temperature of the organelle was assumed to be close to 37 degrees, the temperature of the mammalian body. In fact, when we embarked on this project, we were unsure whether we would even be able to measure the expected temperature differences between, say, 37.4 against 37.1 degrees. Thus, it was something of a shock that our initial measurements implied mitochondrial temperature of 50 degrees Celsius or even greater. Although I don't have time here to delve into all the details of our work and its interpretation, I shall try to outline both the basis of our measurements and the major findings that we've published in these two primary articles illustrated on the slide, which may of course be consulted for further details. I'll come back to these issues in the second and third of these video seminars. First, I think it's appropriate here to chart out the full range of topics that I'll be covering in these video seminars. For the rest of this segment, I'll be coming back to the question of why mitochondrial heat production has been so consistently ignored, why it's been so rarely considered in biology and medicine. In the second part, I'll cover the tools and methods that we've used and can use in the future to extrapolate intracellular temperature. In part 3, I'll address the main points of our 2023 eLife paper and how our findings indicate that mitochondrial temperature tends to revert homeostatically to a little above 50 degrees Celsius in the face of various stresses and disturbances. In part 4, I'll take a bit of a detour through some of our earlier work, mostly in Drosophila, which indicates the biological importance of mitochondrial heat production. In part 5, I'll introduce our speculative ideas on how mitochondrial heat production may impact diverse cellular processes. Then, in the following two segments, I'll look at the specific impact of mitochondrial heat on organism-level processes, first of all regarding evolution, then disease. I'll wrap up by addressing criticisms of our work that have been levelled in part 8, and finally, a whimsical project of my own that I hope will be undertaken someday in Antarctica. Part 10, if it ever happens, will be a follow-up in which we'll address questions raised by you, the online audience. For a long time, mitochondrial heat production was recognized as occurring only in specific physiological or developmental contexts, as a kind of exception to the rule that it was otherwise minimal or negligible. Two famous examples are heat production in the reproductive organs of thermogenic plants, in particular the aroid lilies. Secondly, in the brown adipose tissue of mammals. In both cases, mitochondrial respiration appears to have been repurposed from ATP production to heat production. In aroids, this heat raises the temperature of the flower to over 40 degrees Celsius, enabling the volatilization of attractants for pollinating insects. By uncoupling mitochondrial respiration from ATP production, brown adipose tissue emits heat to maintain mammalian body temperature when body appendages like fur or fat and processes such as shivering are insufficient to combat the effect of environmental cold. This, of course, ignores the fact that mitochondria are already generating the vast bulk of the heat that maintains mammalian body temperature at around 37 degrees Celsius, at least under temperate conditions. Catabolism, the breakdown of nutrient substrates, is an energy-yielding process. But, like any chemical reaction, it's not 100% efficient thermodynamically. Substrate molecules are partially or completely combusted, yielding the ATP that drives useful work such as muscle contraction or nerve conduction, but with a portion always released as heat. Some of the input material is also recycled for biosynthesis. Importantly, the heat is not just some kind of waste product. In homeotherms, such as ourselves, the heat released by mitochondrial respiration, that is, in the business end of catabolism, is what keeps our cells and body temperature tightly around an average of 37 degrees Celsius. But the heat is also important in poikilotherms, like Drosophila, which have wrongly been described earlier as ectothermic, implying that all body heat comes from the outside. Estimates of the energetics of this process indicate that little more than about half of the energy released during catabolism goes into useful work. We should also note, at least in passing, that when ATP is used to perform work, the reactions are again far from 100% efficient, leading to the release of further heat. This even applies to just using our brains, although the data are rather unclear on whether intellectually challenging tasks actually burn more calories than supposedly mindless ones. The ATP synthesized in mitochondria itself may be considered a heat carrier, since its export from mitochondria and then its subsequent hydrolysis in the cytosol is also an exothermic process. It might even be a means to export heat from cells to the rest of the body. The steady state temperature of mitochondria, or indeed of other compartments of the cell, is not determined solely by the rate of heat output arising from mitochondrial metabolism. As shown on the slides, it also depends on the thermal conductivity of the mitochondrial membrane system and also the plasma membrane beyond, plus the heat capacity and conductivity of the cytosol and the extracellular temperature as well. In fact, temperature gradients may exist between cell compartments and across the cytosol itself. Consider the simple analogy of a domestic heating system. Outside, it may be quite cool, while the central heating radiators are warm. But once an equilibrium state is reached, the room is kept at a comfortable steady temperature intermediate between these extremes. Of course, there may be other minor sources of heat inside the room, such as a reading light. Note also that the temperature of the circulating water inside the radiators may be, in fact almost certainly is, hotter than that of the radiator's surface, as determined by the heat capacity and thermal conductivity of the radiator itself. Applying this logic to the cell and to its mitochondria, it follows that the site at which mitochondrial heat is actually generated, i.e. the oxidative phosphorylation or OX-phos complexes of the inner mitochondrial membrane, likely has a higher steady-state temperature than the adjacent compartments of the mitochondria, namely the mitochondrial matrix and the intracrystal portion of the intermembrane space. The temperature of these other subcompartments is not immediately predictable unless we know details of the intramitochondrial architecture and the heat capacities and thermal conductivities of the various membranes and the aqueous compartments that they enclose. In fact, we need to do better than we have done to date and try to measure the temperature of submitochondrial and other subcellular compartments in order to extrapolate experimentally what these heat flows are actually producing. And we'll return to this topic in part 2 of the series. But consideration of these aspects of heat production and flow within the cell have been largely ignored until now, since we didn't until recently have any experimental tools or even theoretical concepts to make sense of them. And that's surely one aspect of why we've ignored the elephant for too long. I'd like to conclude this part of the series by recounting the various other reasons why mitochondrial heat production has been largely ignored. And after that, I'll bring in a contribution from our sister lab in Paris to offer some comments of their own. The structural complexity and variability of the internal architecture of mitochondria has undoubtedly been a further handicap to any meaningful consideration of heat flow within and out of mitochondria, or its measurement, even if we did have the tools. The main focus of mitochondrial bioenergetics for some decades has been elsewhere than on heat production and transfer, most obviously on the molecular marvel of ATP synthase, a miniature rotary turbine. Then, in order for biochemists to standardize measurements, arbitrary temperatures had to be set. Since nobody knew what the internal temperature of mitochondria actually was, the best guess people could come up with was for everything to be done at the standard body temperature of 37 degrees, or, if working with plant or fungal material, some other arbitrary standard temperature. The fact that any deviation from 37 degrees is considered to be abnormal or pathological in medicine strengthened the idea that this was the right temperature at which to do everything. Extreme body temperature anomalies are fatal. Nobody ever survived anything like 50 degrees as an internal body temperature, so doing mitochondrial biochemistry at such a temperature would have seemed illogical and would have been dismissed as non-physiological, and some vestiges of this thinking persist even today. I'll return to this in a later video. Even when things point clearly in this direction, there has been an understandable reluctance to embrace a new paradigm. And if it turns out to be wrong, that would require another handbrake turn in the lab. And of course, we'd have to overturn an entire century of mitochondrial biochemistry, biophysics, and pathophysiology. Here, I'd like to introduce my colleague Pierre Rustin from Paris to say a few words about why he thinks mitochondrial heat production has been so long neglected.
I first studied mitochondrial respiration in heat-producing plants like arum maculatum. After several years on that topic, I set it aside for decades, only to return to it much later when new temperature-sensitive mitochondrial probes appeared. Looking back, it's striking how little attention heat production has received, even though it accounts for most of the energy released during mitochondrial substrate oxidation. This neglect, I believe, arises from both conceptual and technical reasons. Conceptually, bioenergetics has long been dominated by an ATP-centered view, where ATP is the useful energy currency and heat is seen merely as waste. This framework, reinforced by years of chemiosmotic and redox studies, left little room to consider heat as a functional or regulation output. Technically, precise nanoscale thermometry is extremely challenging. Fluorescent probes are sensitive to viscosity and photobleaching, so researchers have focused on easier parameters, ATP, oxygen consumption, membrane potential, rather than elusive temperature changes. Finally, inertia within the field has played a role. It's difficult to link small local heat variations to clear physiological effect, and so the topic remained marginal. To me, it's a mix of entrenched assumption, technical constraint, and conceptual habits that has led to the persistent underestimation of mitochondrial heat production as a key aspect of bioenergetics.
In part 2 of this series, I'll shift attention onto how we've actually set out to measure mitochondrial temperature in cells, saying some words about its limitations and pitfalls, whether the different methods we have adopted give concordant results, and how these methods can be refined in the next steps in the research. The technologies developed by our collaborators now enable us to undertake a series of such steps that will lead us into the once dark world of biological heat flow. It remains for me to say thank you to everyone who's been a part of this research endeavour, notably my long-term scientific partner Pierre Rustin, his close colleague Paul Bény, the postdoc who did lots of the relevant work in my lab, Mugen Terziolu, now a senior scientist who continues the project in the lab of Tiina Salminen, plus the two people who've developed the fluorescent probes that we've used and which I'll describe in more detail next time, namely Young-Tae Chang from Postech in Korea and Takaharu Nagai from Osaka in Japan. And finally, the funders, other colleagues and collaborators who have contributed to our work and our thinking, and the rights holders of the various figures and illustrations I've used in these slides.